any questions this fine morning?
Yes.
Yes, there are questions? Okay, go.
Someone make a question.
Okay.
I'm going to do the urea cycle and be done. That's it.
Okay. For
How long
the
will that take? Quick, no one make a question.
They may
Hour and 15 minutes.
Okay.
We're not going to get to your questions.
for
Go
the
ahead.
for the electron transport chain, the DHAP shuttle to pass the electrons from the NADH in.
Uh-huh.
So that's connected to an FAD that takes them directly to coenzyme Q.
Yes.
Is that different from the other 4 transporters? Or,
Yes, because this one, the other 4 transporters, which are...
well, like the 3 complexes that they go through.
Yes. This is another one, in part because the active site for this one is on the intermembrane space side of the endoplasmic reticulum inner membrane.
Well, how do they decide which one goes to which? Because it's just
Well, all those things where FAD is involved, FAD passes its electrons to Q,
Right.
skipping complex 1.
It always goes. So that's the second. It's just the second complex.
Right.
Okay. Okay.
Does that make sense?
I think so.
Any other questions? Go ahead.
When we were talking about the lipases, so like
Okay.
phospholipase and then pancreatic lipase,
Uh-huh.
we said that phospholipase does DAG, not DAG, diacylglyceride, I think, or something like that.
Yeah,
And then
I may have misspoken there.
And then pancreatic lipase does TAP, triacylglyceride.
Yes. The phospholipase does phospholipids. I think so. That's where I think I misspoke.
Oh, okay.
Phospholipase does phospholipids.
Yes. Okay.
Okay.
And
Those
then
things after I said it, I didn't think that makes sense. Okay.
that's okay. For the Vldl and Chylomicrons,
Uh huh.
are they used differently? Because we said the VLDL is just continually recycled. So why do we need more chylomicrons to come in? Like, do they go to different tissues?
Chylomicron. Where are chylomicrons synthesized?
Well, they go from the small intestine to the liver, but we also said that they still can be sequestered by the different tissues.
Right. So, but that's the way the small intestine introduces absorbed lipids into the system in the form of chylomicrons. So as they are circulating, tissues can extract those lipids if they need them, in the same way that they can extract lipids from the VLDL.
So they're just there at the same time?
Right.
Okay. Does the HDL only go back to the liver? Like, can it be sequestered also by tissues, or is it just a straight shot?
I think it's going back to the liver.
Okay. Squalene synthase?
Yeah? The head-to-head
Yeah.
condensation?
So when is the nucleophile formed? Like, is it before the where is it? Because I was trying to figure it out, and it didn't make sense.
That's okay. I've tried to figure it out. It doesn't always make sense.
That's fine if we want to
no,
stay.
I will not ask you that.
Okay. That's
so
fine.
you're good with that? Yeah.
Uh-huh. Oh, and then we talked about how when we have more cholesterol, we make too much when we have high insulin or a genetic failure.
Yep.
Why is it when we have high insulin? I forget.
Because insulin is signaling that we are in a fed state. And so one of the things that, so insulin initiates a number of things. Glycogen synthesis, right? Remember that sort of, on the glycogen synthase pathway, there was the thing that insulin was involved. So glycogen synthesis, fatty acid synthesis, cholesterol synthesis. It initiates general protein synthesis. So insulin does a number of things.
And that's just one more.
Right, indicating that we are well fed and should make stuff. Right, that's what insulin, and that's why it's the protein synthesis thing. I mean, one of the things, when they talk about performance enhancing drugs, one of the things that bodybuilders take as an inappropriate use of a performance-enhancing drug is they take insulin because it increases rates of protein synthesis.
Can they get in trouble for that?
If they get caught, yeah.
Last one.
Uh-huh.
For the HMG-CoA, we said that there's like the feedback inhibition.
Sorry. Back to the cholesterol.
Yeah.
The other thing we didn't get to is that there's also supposed to be a feedback inhibition of cholesterol on cholesterol synthesis. So another factor in overproduction could be a problem with that feedback inhibition when you've already got enough cholesterol in the system.
Okay.
Okay?
And we said that if we have a lot of the cholesterol, there's feedback inhibition to stop the HMG-CoA. Is it to stop the synthesis of it? Like before so it's before the committed step? It's not the actual committed
The
step?
reductase. The HMG-CoA reductase is the...
Oh, okay.
is
So
the
it is the
committed
commit step.
Yeah. The HMG-CoA reductase is the committed step.
Right, but so it's inhibiting the committed step, not the
Yes.
synthesis.
Yeah.
Okay. Okay.
Yes. Any other questions?
That's all.
Was your banana good? Okay. Any other questions?
Since it is finals coming up, will you be available for email?
Yeah, except James has a double header tomorrow, so that's going to be like 7 hours at the baseball field. Yeah. And one of the things, since I do most things on my iPad now, I've got my personal email routed through my iPad, but not my work email. So I'm not on where I see my work email as much. So I'm going to have to, it's going to be while I'm finishing writing your test. That will be on my computer.
Like a student, like filter out all the Corey potentially and annoying emails and then just get ours.
Yeah. Okay? Any other questions? Because when's our test?
Monday.
Monday, yeah. Monday at 10.
It makes me nervous in my Amanda's class.
I love you, Claire.
So, are we ready to talk about the urea cycle?
Is this going to be on our test?
Yeah.
Oh, we're not just doing it for
Why is
fun?
this different than any other? All right, let's talk about it.
Let's talk about it. Let's get it done.
You just learn for fun. And
Yeah.
We always learn for fun, and then we assess for fun too.
That's my word to use.
Oh, God.
Well, we learn for fun and we assess for fun. So, right, ammonia. We've talked a number of times about using the carbon skeletons of amino acids as raw materials for a number of things. We've talked about using them as raw material for gluconeogenesis. We've talked about using them as raw material to feed into the citric acid cycle to oxidize for energy. Unfortunately, we're not going to have time to talk about the specific metabolism of those carbon skeletons, because there's some really cool chemistry that goes on to take all those different carbon skeletons and break them down into common materials that we can feed into other metabolic pathways. But one thing we can talk about is what happens with the amino group, which can be released in a number of ways, but ultimately is equivalent to having ammonia. Now, ammonia is great. It's really water soluble. Unfortunately, ammonia is also weakly basic, so it affects the pH of the system as ammonia builds up. And we know that biological systems have a limited pH range in which they are functional. But there are some organisms that can just excrete the ammonia. What's the problem with excreting ammonia? Well, one, it's got to build up to high concentrations, and you got to deal with that. The other thing is ammonia is really water soluble, and so it tends to take a lot of water with it. So what kinds of animals tend to excrete their nitrogenous waste as ammonia? Fish. Sarah knew a biology thing.
No, I have goldfish.
Yeah, right? So fish tend to right? Because they live in an aqueous environment. They don't have to concentrate this. They can just let the ammonia diffuse across their gills. And so their gills become an excretory organ, right? So oxygen can diffuse in, ammonia can diffuse out, and so really that's how the fish deal with it. But if you've got to live outside of water, where you can't dilute that ammonia in large volumes of water, which fish tend to live in, right? Then you've got to find some form, some way of making the ammonia both less, making the ammonia have less of an impact on the pH of the system, while putting it in a form that you can store, ultimately concentrate, and excrete using less water, right? Now, for most mammals, that form is urea. And you can see for every molecule of urea, we can get 2 equivalents of ammonia nitrogen incorporated, okay? Urea does take some water to excrete. It is water soluble. And so some water goes with the urea. But this is the form, the major form that we use to excrete our nitrogen. If you were a kangaroo rat, I love my documentaries about kangaroo rats living in the desert when I was a kid. And kangaroo rats are able to concentrate their urine down to where it's almost at the solubility limit of this, so that they can excrete as little water as possible. Now, we're not going to get to talk about this, but uric acid is another form. And you see with uric acid, there's 4 moles of nitrogen per mole of uric acid. What kinds of organisms excrete their nitrogen primarily as uric acid?
Birds.
Birds. Yeah. That white pasty stuff on your car, that's uric acid. Right? What else? What other group is closely related to birds? Reptiles. Reptiles tend to excrete their nitrogen as uric acid as well. So, uric acid, so what do we know about uric acid? Pasty stuff on your car.
No.
It's gross, yeah. Jordan's all over the gross thing. What about the amount of water there?
Not much.
Much less water. So it's a way to excrete that nitrogen and not excrete very much water along the way. Now, does uric acid have an impact on humans? Huh?
Annoyance.
An annoyance when it's on your car. Yeah, I made the mistake one day of parking my car along the building here. That's a really bad mistake. It was covered. Yeah. So, but what else do we have problems with uric acid? You saw it?
Goat.
Gout.
Oh, I thought that was
Looks
a
like
goat.
Gout.
What's gout?
Gout is a disease. It's caused by a genetic problem where we do make some uric acid, but we make our uric acid. What does this look like?
Pure pie.
What? No, not tryptophan.